All right, today we're going to talk about Hardy-Weinberg equilibrium. You might also see it known as Hardy-Weinberg law or Hardy-Weinberg equation, HWE for short. Notice there's the hyphen in the middle. It's because the two scientists, as it were, discovered it independently of one another, so they both get credit for publication. G.H. Hardy was a British mathematician, and Wilhelm Weinberg was an obstetrician or an OBGYN in Germany. So they were both kind of amateur geneticists in their own right at the population level. So a little bit different than the unit we just finished, where we're looking at, genetic crosses between individuals with known genotypes and then probabilities amongst offspring. This is population wide. So what you're really looking for is, or what they realized is that the frequency of alleles in a population, how many dominant recessive alleles of one gene or one locus of a chromosome or genome, all right, it will remain remarkably stable in the event that no evolution is occurring. So why would you want to consider an event like that? Well, you could think of that as almost an ideal, and in seeing how close or far away you are from that ideal, your observed versus expected, or theoretical, versus observed, however you want to phrase that, that'll give you a clue as where to look. How far away am I from what I assumed or what the frequencies should be? And then you can look at a few of these tenets we're about to run through. You'll notice there's 7 of them on your notes. We're going to run through those now. And that'll give you a good idea if you're a biologist or ecologist or anthropologist in the field of where to sort of focus your study. So first, evolution will not occur if mutation is not occurring. Now again, you'll notice as we go through these on the list that none of these are very likely to occur, and that's sort of the idea. We're assuming an ideal condition by which there is no evolution occurring. So again, we know that's not the case, so we could, with that ideal, figure out what is expected, and then from there compare it to what's actually there, and then that'll say, okay, well, which of these things is not happening or is happening, and that'll give us an idea where to focus. So we know mutation always occurs. It could happen from just random mutations, from UV radiation. It could happen from different polymerase errors, for example. So lots of mutations can occur. We talked 2 units ago in our transcription translation unit towards the end about how mutations can occur, carcinogens, viruses, all that stuff. So we know mutations happen. Natural selection is not occurring. Again, we know that this isn't the case. Natural selection is always occurring in any population within an environment. Some individual members of that species will be more or less fit than others. The ones that are more fit have their traits selected for, and then they are going to have a higher frequency of those alleles show up in the next generation. So natural selection is occurring. Right? Is a population infinitely large? Right? If a population is infinitely large, there's going to be enough variation to where no evolution is going to occur. Right? But again, we know that's not the case. Populations are limited by different factors. Predator-prey relationships. You've got the carrying capacity of that environment, different movements into and out of the population. So populations are not infinitely large. Right? Evolution will not occur if all members of the population breed. Well, that's not necessarily the case. We talked about giraffes, we talked about deer, we've talked about a lot of species in class, rams even, to where not every individual member of that population is going to breed. They might try, but if their opposite sex doesn't select for their characteristics or traits, if they can't win those reproductive rights from other males in the population, then they're not going to be able to breed. So we know that that's not true. There are numerous members of every population that never have that opportunity. Next one is all mating totally random? So is that the case? We've talked about selection, a few bullet points ago. So no, mating is not random, at least in the species that we're talking about in terms of this class. We're not talking about all of zoology. There's a lot of ocean critters we're not getting into, but all mating isn't totally random. Okay, evolution will not occur if that's the case. So what type of things happen? Well, you've got individual members of different species are being selected for or against. You have to win the reproductive rights of those females. Look at all the crazy stuff that humans do to attract members of the opposite sex, or that birds do to attract the members of the opposite sex. Look at the colorful displays and the ornamentation that they build around their nest sometimes, or the trinkets or baubles that they will present to the opposite sex in order to win those reproductive rights. So female and another species, sometimes males, will select for different characteristics. So it's not random. Does everyone produce the same number of offspring? Absolutely not. Maybe there's one family with 10 kids whose name all start with J, and who knows, right? Or maybe there's another family with 1 kid and another family with no kids. Who knows? So the same thing's true in our animal cousins. You've got situations where a bear might have 3 cubs, or maybe only 1 or 2 lived through the winter, or you got a situation where there's 1 cub or something like that. So everyone doesn't produce the same number of offspring. Is there no gene flow? Right? So for no gene flow, that would be like the cheetah population we discussed, where there's not enough cheetah genes from an outside population moving into that Great Rift Valley, the savanna there where cheetahs are, the main population we're studying are there. So if there's nothing to move in, you're not ever having an opportunity to weed those inferior genes out. So there is no evolution occurring if it's not new frequency, or sorry, if you don't add to those frequencies with new alleles all the time. So this gene flow one is similar to mutation. New alleles or allelic frequencies can change based on mutations, adding new alleles or versions of a gene that then are expressed as in transcribed and translated into a protein, but also gene flow, just physically bringing in new genes from outside there. For example, if you have one animal that's from southeast Missouri, moves up to St. Charles County and reproduces here, well, you're bringing in new genes to this St. Charles County population. So there is going to have that happen. And again, why do we look at this? This is a situation that's going to occur if no evolution is occurring. Each of these tenets has to be met. If they're not, then evolution is occurring. Now, we just blasted each of these away because all this stuff's happening all the time. We have to think, if we can establish an ideal of what would be expected without any evolution, then you could compare it to what's actually there, and any difference, all right, is that statistically significant or not. So you can do a chi-squared analysis, which unfortunately we don't have time to do, but we will have time to get into Hardy-Weinberg and find out what are these expected allelic frequencies. So this is a simple equation used to track the allelic frequencies from one generation to the next. Remember, the allelic frequencies are important because that's your genotype. The genotype matters. That's, you get half your DNA from mom, half from dad. You're the result of this genetic math, right? Each contributes sperm or egg to our our solution that we are. So 0.5 plus 0.5 equals 1. And you see in any probability, 1 is what you're adding up to. That's the end result. So genotype leads to phenotype, and the phenotype is where we're going to see most of our variation, the expression of those genes. Now we know that variation can be structural, it can be physiological, and it can be behavioral. So don't we already use Punnett squares for this? Like I said earlier, Punnett squares predict the probability of offspring with genome phenotypes based on known parental genotypes. Or even if you're doing a test cross to find an unknown parental genotype, you at least know the offspring genotype already. So this is based on knowns, right? HWE or Hardy-Weinberg equation or equilibrium can do the same thing, but for an entire population. You can look for the expected result of these gene frequencies, which are inherently stable over an entire population in a given area. All of one member of a species in one defined area is your population. So here's the actual equation. I've alluded to it a few times. You see it in your notes already, but p squared plus 2pq plus q squared equals 1. So let's talk about how to apply that. So here's the equation. What you have here is a young woman with albinism, and we're going to use her as an example. So p squared at the end of the day is going to be the frequency of homozygous dominant individuals, as in how many members of this population have the genotype big A, dominant, double dominant, some people call it. We will call it homozygous dominant. 2pq are the heterozygous individuals in a population. Q squared is the frequency of homozygous recessive individuals. How many little a's do you have out there? So you're trying to really find out how many p's and q's you have. How many dominant alleles are out there? How many recessive? Not every little a is in this homozygous recessive genotype. Not every big A is in this homozygous dominant genotype. Some are heterozygotes. So we could do some math to figure that out. The example here, go back to the young lady at the top of the screen and these pink and white polka dot jammies. 1 in every 20,000 humans is affected with albinism or albino. The only way to be albino or have albinism is to be homozygous recessive. So this is an example where we could take a phenotype that has a known genotype and use that as a starting point. So we could figure out what are the predicted frequencies of each genotype, boom, boom, and boom, over an entire population. We're expecting to see 1 in 20,000 little a combos. Let's find out. So how do we do the math? Start with 1 in 20,000. That's as simple as 1 divided by 20,000. That's 0.00005. Now remember, That is our Q squared. All right, 1 in 20,000 people has little a, or homozygous recessive. That's Q squared. How do you get Q from Q squared? You take the square root. You always do the inverse. What you do to one side of an equation, you have to do to the other. So by taking the square root of Q squared, you get 0.007. That's Q. So we know there's 0.007 little a alleles in this entire population. They're not all homozygous recessive. Some of these are actually in heterozygous genotypes, people that are carriers for albinism. So how do you find P? Well, if you know Q, you know it all adds up to 1. P is 1 minus Q. So 1 minus 0.007 equals 0.993. So P is 0.993. That's 99.3% of the alleles out there are a dominant allele, are a normal pigmentation allele. So now we can go back and plug everything into our original equation. So P squared is 0.993 squared. 2PQ, that's 2 times 0.993 times 0.007. And in Q squared, we already figured out. That's right here, 0.007 squared. So do the math, do your order of operations. Q squared is 0.993 squared is 0.986. 2PQ is 0.014. And then 0.007 squared is 0.00005, like I said, like we learned here at the very beginning. All that adds up to 1. So what does this really mean? How do you analyze these results? Well, if P squared is 0.986, that's 98%, 98.6% of the population is homozygous dominant, big A, big A. 2PQ is 0.014, that means 1.4% is heterozygous. And then the last one, 0.005% of our entire population is going to be homozygous recessive. So again, how would you apply this to a population? If you go into an area, say Western Africa, for example, and you're expecting 0.005% of the population to be albino, if you go into a tribe or something like that, or into a village or a small city, and you see more than 1 in 20,000 albinos, if you see a situation where in just one day you run into 5 or 6 or 15 people that are affected with albinism, something's going on. You could go back to the 7 tenets that we wrote down that have to be present for no evolution to occur. You can go back and see, hey, which of those is not in play? All right. Is there more random mutations? No, that wouldn't be likely. Is there a different type of selection going on? That's probably most likely going to happen. You know, artificial selection or even natural selection, but probably artificial in this case. So you could use these numbers to find, hey, this is what I expect to see in a population compared to what you actually see. And then, you know, you have a place. Well, if something is out of whack, so to speak, now you've got 7 things to look at by judging how far away from expected, you know, it actually is. It gives you something to look for in terms of what's going on in this population that's causing it not to be in Hardy-Weinberg equilibrium. And then again, as a biologist or ecologist or anthropologist or anything, it gives you a direction to focus your study. Otherwise, how do you know what data to collect? So the second part of this would be a chi-squared analysis. Once you have your observed, all right, versus your expected, which we just here we figured the expected. Once you go out and actually collect actual data and you've got your observed, how far away does observed have to be from expected for it to be statistically significant? Is it just random chance that that's, you know, the difference you see, or, you know, is something else going on? So there's a part 2 to this that we'll talk about in the future. Now we're going to go through a couple example problems, one with human ears about free versus attached earlobes, and another about PTC, which is phenylthiocarbamide. Some people can taste it, some can't. So we're going to do that in class.